Uh, what is MEN? MEN is multiple endocrine neoplasia part 1. And it's written that Wermer's syndrome, and it is an autosomal dominant inheritance. So they can ask you a question, the other name of a MEN, and that is a Wermer's syndrome. And whether it is an autosomal recessive or dominant, it is an autosomal dominant. A single allele is enough, right? Single allele is enough for the production, autosomal dominant inheritance. Now, this tumorigenesis, this MEN1, okay, the MEN1, this tumorigenesis is at least in 8 endocrine diseases, and it can cause a non-endocrine tissue also. That means if there is any mutation in this gene, okay, any mutation, if there is any mutation which is responsible for this MEN syndrome, right? This not only affects the very specific, is going to affect the 8 endocrines and non-endocrine tissue also. And remember, it is rare, prevalence is 1 in 30,000. If you start screening 30,000 of people, one you'll get an MEN syndrome person. And mostly the age depend is 20 to 40 years of age. Mostly it is 20 to 40 years of age. Rarely if you come across, it is less than 10 years you get a rare. And remember that typically the clinical manifestation of MEN1 will have developed by the age of 29. So if you look into the age, by the age of 29, you might have come across these patients start presenting with the clinical manifestations. So the first point is a Wermer's syndrome, right? Wermer's syndrome, the other name for the men, and this is an autosomal dominant. And this tumorigenesis is at least in the 8 endocrine and non-endocrine tissues. Prevalence is 1 in 30,000. Mostly comes across 20 to 40 years of age, rarely less than 10 years. Typically, the clinical manifestation of MEN1 will have developed by the age of 29. By the age of 29, it might have developed. And these MEN1 families, when I start examining, when start looking for the screening this point, MEN1 families underwent genetic analysis, and MEN1 associated, when I start looking at this point, what happens here is, and this a gene, the MEN1 is localizing, it's a tumor suppressor gene. You need to understand, this suppress the tumor. The MEN1 suppress the, it suppress the gene. And this a gene which suppress, it is present on chromosome 11q13, right? It is present on chromosome 11q13. So now let us understand a few points regarding the genetics of this MEN, multiple endocrine neoplasia. Let us understand where exactly and how exactly. And this genetics of MEN1 is described by the Larson et al. At the year 1997. Chromosome 11q13. You have a chromosome 11q13. And we should also understand 11q13. And we should also understand that it encodes, what does it do? It encodes a 610 amino acids. It encodes a 610 amino acids, that is a nuclear protein, right? Nuclear protein menin. That means this, the genetics of the men is described by the person known as Larson. You have a Larson in the year 1997. And the chromosome here is 11q13. And what does it do? And it encodes, right? That's this gene which is present in the chromosome 11q13 encodes, it has got an amino acids of 610 amino acids, and it gives a protein known as menin. And this tumor suppressor in endocrine tissue A, menin recruits mixed lineage leukemia protein, that is MLL, to the promoters of cyclin dependent kinase inhibitors, that is P18 and P27, right? And it also written that it interacts with the activity protein 1 transcription factor zone and other protein involved in the transcription and cell growth. If you look into the cell cycle of this, you have a G1 phase, right? G0 S phase, G2 phase, and this is metaphase. Now what exactly happen is, Now G1 phase, a cell has to go from the G1 phase to the S phase. Now this a protein menin, it prevents the cell, right, extra growth from the G1 phase to S phase. How does it? This menin, what does it do? It recruits a mixed lineage MLL to the promoter of cyclin dependent kinase inhibitors. The moment it recruits this MLL protein to the promoter of cyclin dependent kinase CDK1, P18 and P27, it stops the transcription and stops the formation of a new cells. And this also interacts with the activity protein 1 transcription factor JUNT and other proteins which are involved in the transcription and cell growth regulation. This interacts with the JUNT and interacts with the, this calls upon the recruits the MLL, that is mixed lineage leukemia protein. It calls to the promoter site of cyclin dependent kinase 1, the P18 and P27, and it also interacts with the protein JUN, and if it stops, it involves in the which transcripts, it breaks down or it stops the transcription on the cell growth regulation. Now say example, you have any mutation in this gene, right, which if there is any mutation in the menin in the gene and the false formation of the menin, so this menin do not recruit this MLL mixed lineage leukemia protein, does not get to the promoter cycle of cyclin dependent kinase inhibitors. It stops, it has got no role in the inhibition, so the cell moves from the G1 phase to S phase and health in the continues, and there is an increased or there is an duplication of the cell or the cancer grows. And we should also understand this, the menin has gotten the functions. It helps in, it helps in the transcription activators, it helps in transcription receptors, cell signaling protein, other proteins. So apart from this, the transcription point, it has a major role, plays an important role in transcription activators, helps in the transcription by CMYB, right, CMYB, and you have got something, you have a transcription report repressors like JUN, and you have a cell signaling proteins CKI. You have got an other protein, okay, other proteins like cell cycle, DNA repair, cell structure. It plays in the other aspects also, right? So that's all you need to understand. Now let us understand how the mutation occurs in MEN1. So if you look into this, the entire gene susceptibility, the important question is the 42% is by the frame shift. And what do you mean by frame shift? That is insertion or deletion of nucleotide, right? What is nucleotide? You have a phosphate, a sugar, and nucleobase. So 42% of this MEN1 is because of a frame shift. A total frame has been shifted. What is the frame? Contains a phosphate, a sugar, and nucleobase. And you have 20 to 25% is missense. What is missense? Where one base is replaced by another base. Say example I've given, I'm not speaking about the example for this man, sorry, I'm speaking for this missense, right, where I'm talking about an example of the missense, not regarding the M1. Missense is, say example, a glucagon is replaced by guanine is replaced by cytosine. When the guanine is replaced by cytosine, the one amino acid which has to, say example, there should be production of alanine. So there is production of, because of C addition of cytosine, you have got a production of other amino acids. So this 20 to 25% is by missense and 14 to 23% is nonsense. And what do you mean by nonsense? Nonsense is nothing but it is introduction, or it introduced the premature and it stopped codon like UAG, UGA. It stopped the codons, so the formation of the formation of the well formed of the menin is stopped. And you have 10.5% of deletion or frame insertion mutation. But the next point is more than 10% of germline MEN1 mutation arise from de novo, right? We don't understand why there is a mutation in the MEN1, right? In the MEN1, why there is a mutation here. More than 10% is de novo, right? Mutation in MEN1 are highly penetrant. 50% of mutant carriers are symptomatic, right, by age 20, and nearly 100% of symptomatic by the age of 60. So if you look into the patients, like 50%, right, by the age of 20%, 50% of the mutant carriers are symptomatic, right? Mutant carriers are symptomatic, and nearly 100% are symptom by age of 60. And how do I explain this? You have something known as two-hit hypothesis. What is two-hit hypothesis? If you look into the diagram here, you got a chromosome, one coming from the maternal side, one coming from the paternal side. You have something known as, there is something as tumor suppressor gene, right? So let me read the point here. Inheritant one MEN1 allele with a gene line mutation, the first hit, and the tumor genesis in specific tissues that occur after a second heterogeneous mutation, the second hit, is acquired in the remaining wild type allele in the single cell. So this is the two chromosome coming from the maternal and the paternal. So this is a tumor suppressor gene. Here in the one gene, say example the maternal gene, there is a mutation in one gene. So one chromosome. When there is a mutation in the one chromosome, there will be an if you look into this, this is the first hit where there is a mutation. First hit, there is a mutation. Next what happens here is the second hit is by the, there's a chromosome loss. So this loss of wild type chromosome, now what happens, there's no function. Now what is remained here, no function of tumor suppressor chromosome, is no function, the tumor is not suppressed here, so the cells moves from G1 phase to S phase. So these are two hit hypothesis. There's inherent of MEN1 allele with the gene germline mutation, first hit, and tumorigenesis in specific tissues that occur after a second heterogeneous mutation. The second hit is acquired in the remaining wild type allele in the single cell. So that's all you should understand regarding the two hit hypothesis. And whenever, say example, a patient has a MEN1, now each patient also has a chances of getting the somatic mutation also, right? Somatic mutations. Now 60%, if you go from above downwards, it can affect the pituitary. You have an anterior pituitary hormones. You have, it can affect, if you go into neck, you have a parathyroid adenomas. If you look into the chest region, you have an insuloma, you have glucagonoma, right? If you look into bronchial carcinoid, right, tracheus, bronchial carcinoid in the lung system. And we should also understand, if you go down above the kidney, you have adrenocortical tumors, right? Adrenocortical tumors. So we should also understand there is something, you have on vipomas, and you have a non-functioning P10 PNETs, okay, PNET, the phosphate and tensine homolog. So the first thing which we need to understand, 60% of the patient who have a MEN1 has gotten glucagonomas, right? 57, bit less, 57% is vipomas, 44% non-functional PNETs, and you have PNETs, 38% is gastrinoma, gastric also, 35% bronchial carcinoid, and you have a parathyroid adenomas, 19% is insuloma, 3 to 5% is pituitary tumors, and 2% is adrenocortical tumors. The highest percentage comes for the 60% glucogonomas. This comes the 57% vipomas. Then what is vipoma? It is an vasoactive intestinal peptide, right? Now 44% is non-functional pigments. So these are the somatic mutations which can also happen in the patient, who have MEN1. Okay, so that's all for now. Let's meet in the next session. Thank you.